everyone. Welcome to the first episode of our new podcast, Crispr Baby. My name is Erin.
My name is Naya.
I'm Jacia.
I'm Cecilia.
I'm Kieran.
If you don't know what CRISPR babies are, they're basically genetically modified babies, human beings. Science is so powerful these days.
Also on our Twitter account at CRISPR_babies. Make sure to follow, by the way.
Nice plug.
Thank you. We asked our followers what general questions they had about CRISPR babies. So for our lovely supporters, we are here to stuff your mind with knowledge.
Our supporter Layla asks, how did scientists even come up with something like CRISPR babies?
It turns out that gene editing can actually be useful when it comes to preventing diseases. Diseases that are carried in the cells of parents can be transported to the baby, and the child could be born with whatever corresponding condition the parent possesses. By using CRISPR technology, scientists are able to manipulate those genes and prevent those cells from basically interfering with the child.
A scientist by the name of He Jiankei, a Chinese biophysicist, was the first to even attempt to use CRISPR technology for HIV, which is an example of one of the many diseases that can be transmitted to a child.
Not even just diseases, but any other type of condition that can be passed down hereditarily.
This includes psychological disorders as well.
Now, audience, you may be wondering, did his experiment work? And the answer is yes.
I believe that his experiment led to the first CRISPR babies to be born. They were twins. If I can remember right, their names were Lula and Nana.
That's correct.
All right, next question.
Our supporter, Tony, asked, how exactly does this CRISPR technology work and how's it used?
Well, to put it in simpler words, a sample of DNA is rearranged and cut. Basically, the cut in the DNA strand will guide certain proteins to a cluster of chromosomes.
Sounds like a very delicate process.
It truly is. Such a scary procedure like many can come with many consequences. This can be very risky and dangerous because those same proteins can cut other parts of the DNA, which can result in major problems for a child and the carrier. One thing that can unfortunately result is the formation of cancer cells.
Yeah, that's very scary.
Many different kinds of proteins can be used for this procedure, though you just have to be very careful.
There is even a debate within the science community about how far you should go with genetic modification.
I know that He Jiankei got a lot of heat from the science community about what he did. Things got so bad that he even lost his job in the process.
He was supposedly breaking the law in his state, but no one exactly knows which law restricts him from genetically modifying the cells.
That's crazy.
Yeah, many scientists are on board with the concept of preventing disease, and others think that it's far too risky.
I think it's a great thing. When done correctly and the scientist has more than enough knowledge on how to use this kind of technology. Oh, and to answer the second part of that question, CRISPR tech is actually used for a lot of things besides preventing diseases.
Oh yeah, one that I find really interesting is designer babies. So apparently with this tech, scientists can be able to genetically modify an unborn child's physical traits like eye color and height, and as well as other characteristics like personality and intelligence.
There's also its role in agricultural engineering. Scientists are able to program certain crops to create twice as much produce, become more durable during extreme weather, and be able to better withstand pests. It's basically like creating designer babies, but with plants, which admittedly isn't as cool.
I agree. But now I think it's necessary that we take the time to say that it's important to do as much research as possible before approaching a procedure like this.
Yeah, definitely.
Uh-huh.
It is very important to look out for the health of others.
Yeah. And next up, a question from Savannah, who asks, is using CRISPR technology on people ethical, which is something I think Naya mentioned earlier.
Oh, yeah.
This question is a bit of a tough one, since everyone has their own opinions on what's ethical and what isn't. But we do know that according to a statement released by the Council on Ethical and Judicial Affairs, genetic modifications on humans to prevent or cure disease is fine, but using it to select traits for any other personal reason is unethical.
This also has to do with the argument that if people were allowed to genetically modify their children, it would create an even bigger division between the wealthy and the lower class, since this type of technology would realistically only be available for people who could afford something so revolutionary and high-end.
Yeah, can you imagine? There's already such a huge gap between us. Adding the fact that rich people will have a distinct advantage in favorable qualities and basically become superhumans would be completely unfair.
Of course, there's always more than just one side to topics like this. Some people have argued that parents have the right to decide how their children will turn out. One supporter of this viewpoint being George Enos, the chair of the Department of Health Law, Bioethics, and Human Rights at Harvard.
Right. These people pretty much state that parents already have a huge amount of control over their kids' lives, especially little ones, like choosing their schools, hobbies, and what they wear, everything. So being able to manipulate their children's genes shouldn't be an issue.
Honestly, it's insane to think about. But we should probably move on to the next question.
Definitely. We reached our last question for our supporter, Giselle, which reads, what does the future have in store for CRISPR tech?
That's a big one. Where do we even begin with that?
Well, we could start off by saying all this tech is still relatively new and holds a lot of potential.
Yeah, Randall Platt, a lead researcher at ETH Zurich in Switzerland, actually tried swapping the Cas9 enzyme for another enzyme called the Cas12a. Cas12a is able to target multiple sites and thus able to edit multiple genes at once, which is something Cas9 can't do.
Oh, and using Cas12a instead of Cas9 also means that genes can be upregulated or downregulated, which opens a lot more possibilities for this technology.
I've also heard that there's been advancements in how the Cas9 enzyme is delivered into the cell. Researchers at Tufts University at the Chinese Academy of Sciences use a lipid nanoparticle to send in the enzyme. This nanoparticle is dissolved in the cell, releasing the contents. Once the contents are inside the cell begins to synthesize its own Cas9 from the mRNA template.
It's a much easier way to deliver Cas9 into the cell to alter its genetic code. Since the enzyme is pretty big and complex, it's difficult to directly deposit into the cell's nucleus.
Interestingly, scientists have even tried using viruses to deliver the Cas9 enzyme into the cell, but it can be very risky since the body might have an immune response to a foreign virus entering it.
That's true. As with everything, the science behind this technology is constantly evolving and improving, so it'll only get more precise and more effective from here on out.
All right, everyone, that's all the time we have for today. My name is Erin.
My name is Naya.
My name is Ajacia.
I'm Cecilia.
And I'm Kieran. Please make sure to follow us on our social media accounts for more CRISPR baby technology info.
Bye! Have a wonderful day!